Microbial Genetics, Scientist Cindy. All living organisms have DNA. This DNA has many of the same basic features in all organisms. Let's first look at some similarities. Then we will look at how DNA can have different features in different types of organisms. DNA has two functions. It has an autocatalytic function, which means that it directs its own synthesis. DNA synthesis is carried out in order to undergo cellular division. DNA synthesis is also called DNA replication. All living organisms use DNA as their genetic material. All living organisms must replicate their DNA in order to undergo cell division. Cell division occurs in order to make new organisms or to replace old, worn-out cells. DNA also has a heterocatalytic function that directs the synthesis of RNA and proteins. Genes are expressed as proteins. Proteins are needed in order to carry out the functions of the cell. All living organisms express their genes using transcription and translation. All living organisms make messenger RNA, or mRNA, from DNA in a process called transcription. mRNA is then used to make proteins in all living organisms. The central dogma of molecular biology states that the information encoded in DNA sequences, known as genes, is expressed as proteins. This process is done in two steps. Step 1 is transcription. Transcription involves making messenger RNA using DNA as a template. Step 2 is translation. Translation is the process that the mRNA sequence is translated into an amino acid sequence to form a protein. DNA and RNA are both polymers of nucleotides. Let's first look at the structure of nucleic acids before we get started. You may recall that nucleic acids include both DNA and RNA. DNA in living organisms is always double-stranded. RNA is single-stranded in living organisms. RNA and DNA are both nucleic acids. Nucleic acids are composed of a string of nucleotides. Each nucleotide has the same general structure, which includes a phosphate group, a type of ribose sugar, and a nitrogenous base. There are some differences between DNA nucleotides and RNA nucleotides. Both DNA nucleotides and RNA nucleotides contain a phosphate group. RNA and DNA nucleotides both contain a sugar molecule. DNA nucleotides contain deoxyribose sugar molecules, while RNA nucleotides have ribose sugar molecules. This is why DNA stands for deoxyribonucleic acid, and RNA stands for ribonucleic acid. There are also some differences between the types of nitrogenous bases that can exist in the DNA nucleotides versus RNA nucleotides. The 4 nitrogenous bases that can exist in the DNA molecule include adenine, cytosine, guanine, and thymine. Note that thymine is found in DNA only and is not found in RNA. The 4 nitrogenous bases found in the RNA molecule include adenine, cytosine, guanine, and uracil. Note that uracil is found in RNA only and is not found in DNA. DNA is double-stranded. These DNA strands are positioned anti-parallel to one another. The DNA in all living organisms are oriented with one DNA strand positioned in the 5' to 3' direction, and the other strand positioned in the 3' to 5' direction. We call this configuration anti-parallel. DNA has a sugar-phosphate backbone. In a molecule of DNA, and RNA as well, individual nucleotides are linked together to make a sugar-phosphate backbone. The bond used in the sugar-phosphate backbone are phosphodiester bonds. Phosphodiester bonds are very strong. Another type of bond found in DNA molecules are hydrogen bonds. In a molecule of DNA, the two DNA strands are connected to each other through hydrogen bonding. These hydrogen bonds occur between the nitrogenous bases that exist on opposing strands. The bases cannot just pair up with anyone. There are base pairing rules. Adenine is only able to pair with thymine. When adenine and thymine pair up, they bond using two hydrogen bonds. Guanine is only able to pair with cytosine. When guanine and cytosine pair up, they bond using three hydrogen bonds. Hydrogen bonds are relatively weak. It is important for the hydrogen bonds to be able to be broken whenever the cell needs to access individual DNA strands. For example, the two DNA strands need to be separated for DNA replication and to make the messenger RNA template during transcription. DNA replication in all living organisms is done in what is called a semi-conservative manner. Semi-conservative means that one parent strand of DNA is conserved in each new DNA molecule. Prokaryotic cells and eukaryotic cells have differences in the shape of their chromosomes. Eukaryotic cells contain multiple linear chromosomes, whereas prokaryotic cells contain one single circular chromosome. There are also differences in extrachromosomal DNA, called plasmids. Some species of bacteria have additional small extrachromosomal circular DNA loops in their cytoplasm, called plasmids. A plasmid is a small DNA molecule within a cell that is physically separated from a chromosomal DNA molecule and can be replicated independently. Plasmids are most commonly found as small, circular, double-stranded DNA molecules in bacteria. However, plasmids are sometimes present in archaea and eukaryotic organisms. Interestingly, plasmids do not carry any essential genes that are needed for normal life processes. Plasmids do, however, alter the sensitivities or abilities of bacteria that possess them. The non-essential genes that plasmids carry are classified as accessory genes. For example, plasmids can carry genes that allow for abilities such as antibiotic resistance, habitat tolerance, and bacterial conjugation, which allows the exchange of genetic material between bacterial cells. There are also differences in where DNA is found in different organisms. In eukaryotic cells, the genetic material, or DNA, is found in the nucleus of the cell. The DNA in prokaryotic cells is found in a region called the nucleoid. Bacteria cells are prokaryotic. They are much smaller than eukaryotic cells. They do not have membrane-bound organelles, and they do not have a nucleus. So the DNA in prokaryotic cells is not in a nucleus. Instead, it is found in a region of cytoplasm that is called the nucleoid. The nucleoid is simply a cytoplasmic location, and it is not a membrane-bound organelle like our nucleus. However, the nucleoid does succeed in packaging the DNA into a relatively small space within the cytoplasm of the cell. Proteins assist in anchoring this genetic material to the nucleoid region. Bacteria are small, and the nucleoid takes up almost 1/3 of the space in the cell. There are great differences between the size of the DNA in eukaryotic cells versus bacteria cells. The genome of bacteria cells are about 50 times smaller than found in eukaryotic cells. In comparison, the eukaryotic genome is huge. Another difference that we see is the fact that bacteria cells do not have histones associated with their DNA molecules. Archaeal cells and eukaryotic cells have their DNA packaged by histone proteins. Also, bacterial cells do not have introns contained in their DNA. Introns are regions of DNA or RNA that do not code for proteins. Archaeal cells and eukaryotic cells do have introns. One of the most striking differences between prokaryotic cells and eukaryotic cells are differences in reproduction strategies. Both prokaryotic and eukaryotic cells can reproduce asexually. Asexual reproduction in prokaryotes is called binary fission. Asexual reproduction in eukaryotes is called mitosis. DNA replication is required for cellular division, including binary fission. Remember that prokaryotic organisms are asexual. This means that they have only one set of genes, so we call them haploid. Two daughter cells are generated as the genetic clones of one parent or one progenitor cell in binary fission. The process of binary fission is less complex than mitosis. First, the prokaryotic DNA replicates. Then, the duplicated strand then moves to another location within the cell, elongating the cell. Next, the plasma membrane then pinches to form a septum. The single cell will separate into two genetically identical daughter cells. Prokaryotes, being asexual, have only one set of genes, meaning that they are haploid. So one single parent cell gives rise to two genetically identical daughter cells. Binary fission is an asexual process, meaning that no genetic variation is introduced. In contrast, eukaryotic cells undergo mitosis. Mitosis is the method by which eukaryotic organisms can undergo a type of asexual reproduction. Remember that eukaryotic organisms have large linear chromosomes. Also, they have more than one chromosome. Replication of DNA is much slower in eukaryotic organisms. Reproduction by mitosis is asexual. Eukaryotic cells are diploid. That means that eukaryotic cells will have one full set of chromosomes from each parent, so two sets of chromosomes total. Mitosis is a more complex process than binary fission. In eukaryotic organisms, the mitotic spindle must form, and DNA replication happens during S phase, which is a phase that occurs long before mitosis even begins. In contrast, binary fission, which occurs in the prokaryotic cell, involves no mitotic spindle, and the DNA replication happens during the first step of binary fission. Eukaryotic cells are also able to undergo sexual reproduction for the formation of gametes, or egg and sperm cells. A diploid eukaryotic organism is formed from the fusion of a haploid egg and a haploid sperm that were formed through the sexual reproductive process called meiosis. Now that we've looked at some of the basic features of DNA and some of the differences of the features of DNA in different organisms, let's now look at the autocatalytic function of DNA. Remember that this is the function in which DNA directs its own synthesis in the process of DNA replication. Remember that DNA replication is done in order for cell division to occur. DNA replication in bacteria is a three-step process. DNA replication in bacteria begins at a single point of origin. This is known as the origin of replication, or ORI. Eukaryotic DNA can be about 50 times larger than the prokaryotic DNA. Since so much more DNA must be copied in the eukaryotic cell, we literally have thousands of origins of replication, or ORIs, all working at the same time to replicate our DNA. So DNA replication in eukaryotes begins at thousands of points of origin. The origin of replication are specific nucleotide sequences located on the chromosome. The ORI is the point at which DNA replication begins. Remember that prokaryotic organisms will have only a single origin of replication. Initiation begins at a specific nucleotide sequence called the origin of replication, or ORI. There are specific nucleotide sequences called origins of replication, or ORI, located on the bacterial chromosome. The ORI is the point at which DNA replication begins. Prokaryotic organisms will have a single origin of replication. Soon after DNA replication has been initiated, a replication bubble will form at the ORI. DNA replication will proceed in both directions or bidirectionally from the ORI. This creates two replication forks. Helicase and topoisomerase I, also known as DNA gyrase, function to uncoil and unwind the DNA. Helicase then acts to break the hydrogen bonds between nitrogenous base pairs. A replication fork is formed when helicase separates the DNA strands at the origin of replication. DNA becomes more highly coiled ahead of the replication fork. Topoisomerase, or DNA gyrase, breaks and reforms DNA's sugar phosphate backbone ahead of this replication fork, thereby relieving the pressure that results from supercoiling. Single-stranded DNA binding proteins will then bind to the single-stranded DNA to prevent the double helix from reforming. Now let's look at the elongation step in DNA replication. DNA polymerase can only synthesize DNA in the 5' to 3' direction. DNA is double-stranded, with one strand oriented in the 5' to 3' direction, and the other strand oriented in the 3' to 5' direction. The consequence of this anti-parallel configuration, coupled with the factor that DNA polymerase can only synthesize DNA in the 5' to 3' direction, is that one of the DNA strands will be replicated continuously, but the other will not. The lagging strand of DNA is the strand of DNA double helix that is oriented in the 5' to 3' manner. Therefore, its complement must be synthesized in a 3' to 5' manner. But here's the problem. DNA polymerase III cannot synthesize in the 5' to 3' direction. So, this lagging strand is synthesized in short fragments known as Okazaki fragments. How this is done is through primase building RNA primers to allow for this to occur. Primase builds these RNA primers. Then, DNA polymerase uses the RNA primers to synthesize DNA in the 5' to 3' direction. The leading strand is the strand that is replicated continuously by DNA polymerase. The lagging strand is the strand that is replicated discontinuously by DNA polymerase. DNA polymerase is the enzyme that is required for DNA synthesis or DNA replication in all living organisms. However, there are many different types of DNA polymerases in different organisms. In bacteria, it is DNA polymerase III, or DNA Pol III, that adds individual nucleotides according to their complementary bases. For example, adenine would be the complementary base of thymine, and cytosine would be the complementary base of guanine. DNA ligase is then used to join the Okazaki fragments together. DNA ligase then joins Okazaki fragments together. It does this by joining deoxyribonucleotides together, completing the synthesis of the lagging strand. DNA ligase is the enzyme that functions to connect the Okazaki fragments together for the copy of the lagging strand. The word ligate in Latin means to tie together. Termination of DNA replication. Since the bacterial genome consists of a single circular chromosome, at the end of the elongation step of DNA replication, you actually will end up with two concatenated or interlocking circles of DNA that must be separated. Here are the steps of termination of DNA replication in bacterial cells. Bacterial topoisomerase 4 induces a double-strand break into each of the DNA molecules, allowing them to be separated from one another. In other words, to become unconcatenated. Bacterial topoisomerase 4 then reseals each DNA molecule back together again, creating two separate unlinked chromosomes. Next, we'll talk about the second function of DNA, which is the heterocatalytic function of DNA. This is the part of DNA that directs the synthesis of RNA and proteins. This is the means by which genes are expressed as proteins. These proteins are needed to carry out the functions of the cell. This process is also known as the central dogma of molecular biology. Genes are segments of nucleic acids. Genes are segments of nucleic acid sequences that code for proteins that exhibit a specific genetic trait. Genes are expressed as protein through a two-step process. Step 1, transcription. Step 2, translation. The central dogma of molecular biology explains how information encoded in DNA sequences, or genes, is expressed as proteins in this two-step process. In step 1, we have transcription. Transcription involves making a messenger RNA molecule using DNA as a template. Step 2 is translation. Translation then translates the messenger RNA sequence into an amino acid sequence to form a protein. In short, transcription involves using the DNA molecule as a template to form messenger RNA. Eukaryotic cells and archaeal cells contain non-coding regions of DNA and RNA called introns. These introns are removed from the mRNA through RNA processing. Also, remember that eukaryotic cells have a nucleus. This means that the process of transcription will take place in the nucleus of eukaryotic cells. Prokaryotic cells do not have a nucleus, so the process of transcription will take place in the cytoplasm or the nucleoid region of the cytoplasm in prokaryotic cells. The second and final step of this process is called translation. Translation is the process by which the messenger RNA sequence will be translated into an amino acid sequence which makes up a protein. Keep in mind that we are using the same names in 3 different processes here. DNA replication, transcription, and translation all have the same names for their processes: initiation, elongation, and termination, but they are definitely not the same. Let's talk transcription. Transcription in bacteria is a 3-step process. RNA transcription in bacteria is done in the cytoplasm, not in the nucleus like in eukaryotic cells. Transcription in bacteria is done using an enzyme called RNA polymerase. RNA polymerase adds nucleotides according to their complementary bases, using a free 3' hydroxyl group of the growing nucleotide chain. RNA transcription requires a promoter. A promoter is a DNA sequence that transcription factors bind to. Promoters are located upstream from the sequence that codes for proteins. The nucleotides that code for proteins would be considered to be located downstream from the initiation site. The initiation site is where transcription begins. Promoters usually are located between -10 and -35 bases upstream from the initiation site. The initiation site is designated as +1. All species of bacteria have 2 nucleotide sequences that have been conserved. These are known as consensus sequences. The 2 consensus sequences are as follows. The -10 consensus sequence is called the Tata box. The Tata box has the sequence TA-TA-AT. The -35 sequence is recognized and bound by the sigma subunit of RNA polymerase. Once all of the relevant transcription factors have bound to the promoter region, elongation can begin. In the elongation phase of RNA transcription, the sigma subunit of RNA polymerase falls off of the promoter, and this allows RNA transcription to begin. The RNA nucleotides are added in a complementary fashion to the DNA template in the 5' to 3' direction. In the bacterial cell, this occurs relatively quickly, approximately at a speed of 40 nucleotides per second. And remember, the complementary base pairing rules are AU in RNA instead of AT. CG are still the same. This is because RNA contains uracil instead of thymine. RNA does not have thymine, so when the DNA template contains a nucleotide with an adenine base, it will need to add a nucleotide with uracil instead. RNA transcription is terminated when the bacterial RNA polymerase dissociates from the DNA template and releases the newly made RNA. This is accomplished through a terminating sequence. This is a repeated nucleotide sequence that signals to the RNA polymerase to terminate the process. After transcription, but before translation, specific regions of the primary RNA transcription have to be spliced out in eukaryotic cells as well as archaeal cells. Bacteria cells, though, do not have this issue because they do not have these non-coding regions called introns. These introns are spliced out or cut out and degraded during RNA processing. On to step 2, translation. Translation in bacteria is a 3-step process. In translation, the sequence of bases in the messenger RNA molecule are converted or translated to an amino acid sequence that will make up a protein. Ribosomes function to catalyze this translation of the messenger RNA sequence into a protein. Ribosomes are the site of protein synthesis, but they do not actually make the protein. Initiation of translation. In bacteria, translation of messenger RNA into an amino acid sequence begins with the formation of an initiation complex. The initiation complex is formed when the messenger RNA, the transfer RNA, and the first amino acid all come together within the ribosome. Elongation of translation. The nucleotide sequence of messenger RNA is read in triplets called codons. All translation begins with what is called the start codon. The nucleotide sequence of messenger RNA is read in triplets called codons. All translation begins with what are called the start codon, which is AUG. All translation begins with what is called the start codon, which is AUG. This means the first three bases on the messenger RNA are adenine, uracil, and guanine. The AUG codon codes for the amino acid methionine. Termination of translation. Termination of protein synthesis occurs at the stop codon. There are three different codons which are considered stop codons. This includes UAA, UAG, and UGA. When one of these stop codons reaches the ribosome, the last amino acid will break free from the ribosome and from the transfer RNA. The protein is now complete.